Hello first year students, now we are going to prepare nutrient broth and nutrient agar. Okay, this is the label you can see is of peptone. Okay, and we have already weighed peptone, that is 1 gram for us with the help of this weighing machine. Okay, then after the second ingredient we have is yeast extract. Okay, now yeast extract is already weighed and quantity is very less, that is 0.3 gram, and then after we have to take sodium chloride and that is also weighed exactly 0.5 ml, 2.5 gram. Okay, now with the help of this measuring cylinder, this is measuring cylinder, and we are going to take the water. Okay, so we will exactly draw distilled water 100 ml. Okay, so now it is exactly 100 ml. Now I will take a flask. First of all I will add peptone in it. I will add peptone in the flask. Okay, then after I will add yeast extract, then I will add sodium chloride, and now I am going to add distilled water. Okay, I am adding distilled water. So now after that we have to shake this very nicely. Okay, shake it properly till the ingredients will be mixed up, and if it is not being shaken properly you can use any spatula or pipette or anything for just mixing it. Okay, now this is our medium which is already now mixed. Now with the help of pH strip, I have told you that we are going to measure the pH. Okay, so for that we should take our neutral pH strip. And with the help of this strip we are going to get the exact pH. Okay, so let me do it by myself. I will take out one strip of pH, and now I am going to get some material out of it and I will check the pH. Okay, now I am having no difference in the color. Okay, the color is the same one. Now I will try to see the result. Okay, so this is my calibration. As I have told you that you should require calibration, and you can see that pH is exactly 6.5. Okay, now what is to be added? Now little alkali, that is NaOH, is to be added now. Okay, so here we have NaOH and we are going to add a little drops of NaOH in it. Okay, so two drops of NaOH, 2 to 3 drops of NaOH is sufficient to make the pH alkaline. Okay, now again we will see that what is the difference in the pH. Okay, now shake it properly, the flask, and then check the pH. Now you can see that exactly the pH is 7. Okay, can you see the pH 7 exactly? Yes. Okay, so now our pH is 7. There is no need in it, it may be in between 7 to 7.5. So our pH is adjusted now. Okay, it is already adjusted now. So now we are going to distribute this nutrient growth. Okay, if we want to have nutrient growth, we will distribute in test tubes, but if we want to make agar, then we are going to add agar powder. Okay, this is our agar powder, you can see. Okay, agar powder, and it is to be added in the quantity of 3 grams. Okay, so you can add agar powder accordingly. If we have taken 100 ml of distilled water, you are going to weigh 3 grams of agar powder, and then after you can add it in this. Now before sterilizing it, if you want to sterilize this flask altogether, then you can sterilize, you can plug the mouth of the flask with cotton wool, and then you can sterilize it. Okay, so that is all. Thank you very much.